This experiment, conducted by Michelson and Stahl, elegantly demonstrated that DNA replication is semi-conservative. The E. coli bacteria were first grown in heavy nitrogen, N15, making their DNA denser, switching to normal nitrogen, N14, and allowing the bacteria to replicate, once resulted in DNA with an intermediate density, seen here in the centrifugation results. This proved that each new DNA molecule inherits one original parental strand and one newly synthesized strand. As we see in the diagram, the double helix unwinds, and each strand acts as a template for building a new complementary strand, perfectly illustrating the semi-conservative mechanism. Bacterial DNA replication begins at a specific site called the origin of replication, or ORI. The DNA double helix unwinds here, forming a replication bubble. Notice the two replication forks moving in opposite directions. This is what we call bidirectional replication. At each fork, DNA polymerase synthesizes new DNA. The leading strand is synthesized continuously, while the lagging strand is constructed in short segments called Okazaki fragments. Replication proceeds until the two forks meet at the terminus region. Eukaryotic DNA replication, while similar to bacterial replication, involves a more complex initiation process. The origin recognition complex, or ORC, binds to the origin and recruits other proteins like CDC6 and CDT1. These, in turn, load the MCM complex, which acts as the helicase, unwinding the DNA. Now, look at the key players. DNA polymerase synthesizes new DNA, primase creates RNA primers, topoisomerase relieves tension ahead of the fork, and ligase joins Okazaki fragments. This coordinated effort ensures accurate duplication of the genome. Let's zoom in on the action at the replication fork. See how the leading strand is synthesized continuously in the 5-3 direction? The lagging strand, however, is synthesized in short Okazaki fragments, also in the 5-3 direction, but moving away from the fork. Each fragment requires a primer laid down by primase. This difference arises because DNA polymerase can only synthesize in one direction, and the two DNA strands run anti-parallel to each other. This slide highlights the essential role of the sliding clamp, pCNA, in eukaryotic replication. pCNA encircles the DNA, tethering DNA polymerase delta to the lagging strand and polymerase epsilon to the leading strand. This enhances the processivity of the polymerases, allowing them to synthesize long stretches of DNA without detaching. We also see other essential enzymes like primase, helicase, and DNA polymerase alpha, which initiates DNA synthesis. Finally, RNase H removes the RNA primers, and ligase seals the gaps between Okazaki fragments, creating a continuous DNA strand. The cell cycle is the series of events that leads to cell growth and division. It consists of four main phases: G1, S, G2, and M. During G1, the cell grows and prepares for DNA replication. In the S phase, DNA replication occurs, duplicating the genome. G2 is another growth phase preparing for cell division. Finally, the M phase encompasses mitosis, where the duplicated chromosomes are separated, and cytokinesis, where the cell physically divides. Cells not actively dividing can enter a quiescent state called G0. Cell cycle checkpoints act as quality control mechanisms, ensuring each phase is completed correctly before proceeding. We have three main checkpoints: G1, G2, and M. These checkpoints are regulated by cyclins and cyclin-dependent kinases, or CDKs. Specific cyclin-CDK complexes are associated with each checkpoint, ensuring the cell is ready to transition to the next phase. If problems are detected, the cycle can be halted to allow for repairs, or, in severe cases, trigger cell death. Cyclins and CDKs are the master regulators of the cell cycle. Different cyclins are expressed at different points in the cycle, as seen in this graph. These cyclins bind to and activate CDKs, which then phosphorylate target proteins, driving cell cycle progression. This cyclical rise and fall of cyclin levels ensure the orderly progression through the cell cycle phases. External growth signals, called mitogens, play a crucial role in initiating cell division. Mitogens activate signaling pathways that lead to the expression of cyclin D. Cyclin D then binds to CDK 4/6, activating the complex. This activated complex phosphorylates the retinoblastoma protein, PRB, releasing the E2F transcription factor. E2F then triggers the expression of genes needed for DNA replication and S-phase entry. CDK inhibitors, or CDKIs, can act as a brake on this process, preventing uncontrolled cell division. Targeting CDK2, a crucial regulator of the S-phase, is a strategy employed by some anti-cancer drugs. Flavopiridol, for example, inhibits CDK2, effectively blocking DNA replication and halting cell cycle progression in cancer cells. This slide shows how the molecule interacts with CDK2, preventing it from carrying out its function. Mismatch repair systems, such as base excision repair, or BER, is a crucial pathway for fixing minor DNA damage like single damage bases. There are two main subpathways: short patch BR, which replaces only the damaged base, and long patch BR, which replaces a short stretch of nucleotides surrounding the damage. The process involves DNA glycosylases, recognizing and removing the damaged base, followed by the action of AP endonucleases, DNA polymerase, and DNA ligase to complete the repair. Nucleotide excision repair, or NER, tackles more significant DNA lesions like thymine dimers caused by UV radiation. The process begins with damage recognition by specific protein complexes. The DNA is then unwound around the damaged area, and an endonuclease cuts out a short stretch of DNA containing the lesion. DNA polymerase fills the gap, using the undamaged strand as a template, and ligase seals the nick, restoring the DNA's integrity. DNA repair pathways such as mismatch repair and excision repair share some fundamental steps. First, the damage is recognized. This often involves a change in the DNA structure. Then, enzymes excise the damaged portion. DNA polymerase fills the resulting gap, using the undamaged strand as a guide. Finally, DNA ligase seals the remaining nick, completing the repair and restoring the DNA to its normal state. Double-strand breaks are a severe form of DNA damage and require specialized repair pathways. Non-homologous end joining, or NHEJ, involves the direct joining of the broken ends, while quick, NHEJ can introduce small errors. Homologous recombination, or HR, is a more precise pathway that utilizes a homologous DNA template for accurate repair. These pathways are not only crucial for maintaining genome integrity, but also play a role in gene editing technologies, allowing for precise manipulation of DNA sequences.